Okay, in this lecture video, we're going to talk about some roles for carbohydrates other than as fuel sources. So even though that's their primary function, they have a lot of other jobs. And some of those other jobs include things like structural support. So roles in, for example, peptidoglycan and the outer cell wall of bacteria. We see a lot of carbohydrates even in our own cell membranes, serving structural roles. They're important in cell signaling. I can't spell signaling, so I'm writing signals between cells. So they carry information and allow cells to communicate with the extracellular matrix and hence to each other. And in this role, they're often linked to other biological molecules. So we're going to talk about carbohydrates linked to proteins, and we're going to talk about carbohydrates linked to lipids, which are fat molecules, and coming up later in chapter 10 and 11. But let's look first at carbohydrate protein macromolecules. And we can sort of break that down into two different parts. We can break that down into molecules that are mostly carbohydrate and a little bit protein, and mostly protein with a little bit carbohydrate. So here in this example from figure 724, I've got two examples. So our first is a proteoglycan, which I managed to cut the top of this one off. So this one right here is a proteoglycan. So what we're looking at here is a molecule that's got a core protein, which is what's shown here in pink, this little part right here, but then it's mostly carbohydrate. So what we've got is a lot of carbohydrate up here, and these carbohydrates then serve the roles mostly of grabbing ligands and modulating their interaction with receptor proteins here on the cell surface. And what's really cool about this is proteins can control the amount of sugars in those carbohydrates attached to the protein backbone, and hence they can control how much of a barrier there is to proteins trying to get up next to the cell surface and interact with it. So let's take a look at an example here. So this is an example of an agarcan, and what you're seeing here is along this main line down the middle of this macromolecule, that is a series of repeating disaccharides. So the core structure of this is sugars that are linked together. And then coming along the sides here, we have the agarcan core protein. And again, coming off of that are these chondroitin sulfates, keratin sulfates. So these little molecules here and here, which are again more sugar molecules. So agarcans are molecules that interact with the collagen in cartilage. They're highly solvated. So we have all these sugars on there, all these hydroxyl groups, which you can interact with the water molecules. And this contributes to the development and strength of the tissue. And the extracellular matrix is full of these. So it really can serve for signaling between cells in multiple directions. So here's another picture from your book sort of representing that. Here's these proteoglycans out here in the extracellular matrix. And you can imagine that if you change how much of these there are, then you're changing access to this cell surface. Okay, so proteoglycans have a core protein, which is anchored typically to the cell surface, but then there's lots of sugars, so we call those glycosaminoglycans. So they're covalently linked sugars to the protein backbone. So the second example then that we want to talk about are glycoproteins. So these guys right here. So now glycoproteins are mostly protein. So they're more protein, but they've got sugars attached to them now. And these sugar groups are attached typically through serine and threonine side chains, through the OH group. We call those O-linked sugars, or they're attached through the asparagine side chain, and we call those N-linked sugars. So let's take a look at those. So in this picture, you can see, here's a glycnac, that's my favorite sugar. So you can see the sugar, and here it's linked. We have a substitution at the hydroxyl group on that anomeric carbon, and that's attached to the nitrogen of the amide side chain of an asparagine residue. Whereas up here for this GalNAc, now we're linked instead to the hydroxyl group of a serine amino acid. So remember, in these cases, for glycoproteins, we've got mostly protein with a little bit sugar. And functionality, if you consider the fact that you can, again, vary the length of the sugars, so how many sugars are attached. You can vary the location of where those sugars are attached, and typically they're amino acid sequences which serve as target sequences for glycosylation, which means the addition of sugars. And then you can change which sugar units you have and the order at which they're attached. You can get a lot of variation. So you can take a protein, which already can have lots of variation in it because we have 20 amino acids to play with. But now you can add all of these different carbohydrate functionalities and get a really unique structural surface that, again, lets you have lots of specific interactions. This can affect, physiologically speaking, it can affect solubility, right, of your protein. So you could take a protein that may not be as soluble, cover it with sugar groups, and now it will solvate fine. It might affect folding. So having those carbohydrate arms on there might help it assume its three-dimensional functional structure. It could serve as protection. You can imagine if you have all of those sugars attached to your protein, it would be difficult for other proteins to get close. And oftentimes, those attached sugars might signal that particular protein for degradation. So for example, ubiquination, the addition of ubiquitin molecules, which are sugars, or actually ubiquitin is a protein, but we can talk, that does the same purpose. As you add these sugar molecules to your protein, the body recognizes that as, hey, we need to break this protein down and turn it over. And this is a big deal. A lot of times when you're trying to isolate or express proteins in the lab, for example, by expressing them in bacteria, you can't add these sugar groups because E. coli are prokaryotes. They don't have the ability to do that post-translational modification and add sugars on. So you start making these proteins without their natural sugars. So whether or not that's important for function is something to consider. Of course, at the same time, once you put all those sugars on there, you end up with a very heterogeneous population of proteins to work with. So that's proteoglycans and glycoproteins, which differ in the ratio of protein to carbohydrate. Now we can look at our third other class of molecules that are glycosylated, and that's lipid molecules. So in this case, here's our this is an example of a glycosphingolipid. So now we're talking about attaching sugars or carbohydrates to lipid molecules which compose the cell membrane. So some really good examples of this would be gangliosides, which have sugars for a top, essentially, so sugars attached at the head group. These are important for blood typing. And another important consideration is LPS, which is lipopolysaccharide, which is a component of the bacterial cell wall. So here's an example. So you've got these O-chain linked sugar molecules. We've got the core structure here. And then we've got a molecule called lipid A, essentially. Oh, that one's already labeled. I don't need to label that one. Oh, I guess everything was already labeled. Oops. Okay, so this is LPS, which is a component of the bacterial cell membrane, and also a highly recognized antigen by our body. So this is toxic to us, but our bodies recognize it to start an immune response. So there are lots of classes of these different molecules, lots of different lipid and protein carbohydrate molecules that play lots of roles in the bodies, and you can read about these if you're interested. But from a structural standpoint, I think we've talked enough about the general structures of carbohydrates, how they're parts of other molecules, and their roles, at least in terms of structure and signaling. You'll see their role a lot if you take metabolism, since these are going to be the primary fuel of the body. So the last thing I want to talk about is characterization of these molecules, which is not trivial. Okay, so when we're talking about studying carbohydrates, if we want to characterize them, there's a lot of things we got to figure out. So the first thing we got to do is figure out composition. So if you've got a complex carbohydrate, you've got to figure out, first of all, what what monosaccharides are present. Very similar to figuring out with a protein what amino acids are present. So this is typically done by hydrolysis, and there are also enzymes that will specifically release different sugar molecules, and then followed by some sort of chromatography so that you can actually identify what those molecules are. Then the next step is to get the sequence of those monosaccharides, because we said you could add these together in lots of different orders, just like you can amino acid. And like I just mentioned, there are enzymes that will specifically recognize different sequences of monosaccharides that are polymerized, and so you can release carbohydrates in specific ways, much again like proteolysis allows us to figure out amino acid sequence. So that's great, getting composition and sequence, but that's not all we need to know. So the next thing we would need to know would be that's a 3. Oh, maybe I should just write it neater. Would be the location of the actual glycosidic bonds. So for example, knowing you have a disaccharide made from glucose and fructose in that particular order is great. But you also need to know which carbons are involved. Do you have any branch points? And that's typically done through methylation experiments, where you can block free hydroxyl groups and then figure out by what's left over which hydroxyl groups were instead participating in glycosidic bonds as opposed to being free. So this gives you the types of glycosidic bonds, the carbons that are involved, and whether or not you have branch points. Then the last thing, right, so now you know the glycosidic bonds, but we also need to know the stereochemistry, right? Are we talking about alpha or beta glycosidic bonds when appropriate? So this stuff is pretty tough, these things. So we're usually talking about NMR and mass spec experiments. So it's no longer just about running gels and things like that. So it's going to take NMR, high resolution NMR and high resolution mass spec to get which carbons are participating in these glycosidic bonds and what their stereochemistry is. So hopefully you can see why studying carbohydrates, especially complex carbohydrates, is a little bit more involved than it is to just get basic information about a protein structure. Okay, that's it.